Hello friends, welcome back to the second video of the cells of immune system. Myself Febi Paiva, Assistant Professor, Department of Zoology, St. Joseph's College for Women, Alapura. The first video we have discussed with the different types of cells of immune system. The first type, it was the cells of innate immunity, and we discussed with the neutrophils, we discussed with the eosinophils, basophils, the mast cells. Now we may continue with the next cell which is responsible for innate immunity is the monocyte. Monocyte, we can see that they are partially differentiated cells, that is, once formed within the bone marrow from the progenitor stem cells, they enter the blood system and they remain in the blood for about 8 hours. But then they are in a partially differentiated state. That is not their final stage. And we can see that within when they undergo this transportation through the blood, they undergo certain changes. They in fact enlarges in size, and they happens to go and settle in the tissues where they completely mature or differentiate into macrophages. So when we speak about the monocyte, they are agranulocytes and they do have a bean-shaped large kidney-shaped nucleus. And we can see that they are also involved with phagocytosis as well as intracellular killing of a microorganism. And when they are converted to a macrophages, they undergo certain changes, like we have said before, they enlarge in size. That is, they enlarge in size about 5 to 10 fold. There is an increase both in the number as well as complexity of the organelles. The third one is we could see that there is an increased production of the phagocytic intracellular lysozymes, and we can say that there is a great magnitude of phagocytosis too when they become macrophage. So this is the macrophage, and we can see that the size have increased, the number of cell organelles also have increased, the amount of lysozyme production will also be high, and so there will be a larger magnitude of phagocytosis. So these we can see that they are phagocytic nature, and even they are larger than the neutrophils. And they have a very long lifespan and they can remain for years in the tissue when compared to the monocyte, which just had 8 hours of survival within the blood. These macrophages can be found in different organs like liver, spleen, lungs, kidney, lymph nodes. Both the monocyte and this macrophage are antigen processing and antigen presenting cells. And when we speak about the activation of this macrophage, for the activation, they need a stimulus from some chemical mediators, like one is the endotoxins produced by the antigen itself, or the second one is the chemicals which are released by the T cells, that is the cytokines. So these chemicals give informations to this phagocytic macrophage to become active. So once they become activated, they become phagocytic and they kill the microbes. And when they carry out the digestion of the microbes, they present the antigen to the T cells for their activation. So this is by the same time antigen processing as well as antigen presenting cells. When we speak about the types of macrophages, based on the position in the organs, they are of different types, like the macrophage which is seen in the lymph node, second one the alveolar macrophages which are seen in the lungs, third one the macrophages which are seen in the liver, which are also known as Kupffer cells, and the fourth the macrophages which are seen in the spleen and the bone marrow. So the various names of the macrophages in different tissues are like the alveolar macrophages for the lungs, histiocytes for the connective tissues, Kupffer cells, the macrophage which are seen in the liver, mesangial cells for the macrophages of the kidney, microglial cells for the macrophages which are seen in the brain, and the osteoclasts are the macrophages seen in the bones. Now we move on with the next cell, that is the dendritic cell. This cell is also responsible for the innate immunity. And here these are also phagocytic cells and are found in most of the tissues. By birth, by origin, they can be originated either from the myeloid lineage or from the lymphoid lineage. And they are normally seen in large number at the interfaces of areas which are coming in contact with the external environment, like for example the skin, the mucus layer of the gastrointestinal tract, etc., and where we believe that those are the entry point for the pathogen. So these cells are normally confined to those areas which are coming directly in contact with the external environment. And by nature, these are actively motile. And as they move on, they take the samples of the surroundings by the process of endocytic process, and thereby they recognize the pathogens. These are also efficient in activating T cells, and also they are more potent antigen presenting cells when compared to the macrophages and the neutrophils. There are different types of dendritic cells, like the Langerhans cells, the interstitial dendritic cells, the myeloid cells, the lymphoid dendritic cells, based on the position where they are being seen. So when we speak about the normal dendritic cells which are seen in the skin, they are normally known as Langerhans cells. These are normally derived from the bone marrow, and they are involved in the T cell stimulation. They process the antigen but cannot present it to T cells. What they normally do is they pick antigen from the skin. These Langerhans cells, they normally do not directly present it to T cells, but then what they do is they take the antigen from the skin, they carry the antigen via afferent lymphatic vessel, and now they reach the lymph nodes, where the dendritic cells are known as the wild cells. And here in lymph nodes, the dendritic cells, known as the interdigitating cells, they present the antigen to the T cells. So here we can see that the Langerhans cells, they pick up the antigen, and it is carried via the lymphatic vessels, and they reach the lymph nodes. And within the lymph nodes, they are being presented by with the help of the cells, dendritic cells known as the interdigitating cells. Next is something known as the follicular dendritic cells, which are not having the same origin as that of the other dendritic cells, but then they have given that dendritic name because of its morphology. So they are having a different origin, at the same time a different function. They, as I said earlier, they do not origin from the bone marrow, and they also have a different function, and they do not express the MHC class 2. So they do not present the antigen to the T helper cells. Then how they bring about the action is that they display a receptor, that is a antibody receptor, IgG receptor, and C3 complement receptors, and they entrap a antigen which is being formed a complex which is present as a complex like antigen antibody and the C3 complement complex. And once such a complex is formed, it is bound to this follicular dendritic cells, and they now present it to the B cells, which helps in the B cell activation and thereby the antibody production. So they are, this follicular dendritic cell, they are not concerned with the T cell activation, but then they are concerned with the B cell activation. Next is the null cells. These are also known as large granular lymphocytes. About 15% of the circulating lymphocytes are the null cells. Here we could see that this null cells, they encounter the pathogen in a non-specific manner. That is, they do not recognize foreigner antigens presented on the target cell. They also originate in the bone marrow, and they do not have a specific receptors as we see in other lymphocytes. Since there is no specific receptors, we call them as null cell. The null cells, there are different types of null cells. The category include natural killer cells, antibody dependent cytotoxic cells, lymphokine activated killer cells, LAK cells. So we'll start with the first type of null cell, that is natural killer cell. So the major portion of the null cells belongs to this category. They are normally cytotoxic, and they normally kill the population which are virally infected or tumor uh, type of cells. They do not kill microbes directly, but then they kill the cells in which microbes are infected. That is, they kill the damaged or infected cell, and they do not directly encounter the pathogen. And here we can see that how they bring about such a reaction is that these natural killer cells, they have a particular specific type of receptor known as killer inhibitory receptors. And with this receptor, they access the one of the receptors seen in all the cells in our body, that is all the nucleated cells in our body. We call it as MHC class 1. It is a receptor. So using the KIR, that is killer inhibitory receptors, which is seen on the natural killer cell, using that receptor, they recognize another receptor on the other cells, normal cells. And those cells which are having that MHC1 class receptor, those are not killed by these cells. But then they kill or they entrap those cells which do not have that MHC class 1 receptor, because we know that the damaged and the cancerous cells do not have the MHC1 molecule on their surface. So since they do not have that MHC1 receptor, this natural killer cells happens to kill them. And how they kill is that they kill the infected cell by creating holes by perforins, a chemical which is released by them. They also release another protein, granzymes, which enter through the spores. Once the pores are formed by the perforins, these granzymes, they enter through the pores and they initiate apoptosis and kill the damaged or the infected cells. So they happens to find, they do not require any activation or any confirmation to kill those cells which are not having the MHC1 molecule. Next one is antibody dependent cytotoxic cells. They express Fc receptors, and these Fc receptors will recognize the Fc part of the antibody. And once they normally binds to the antigen which is bound to the antibody. So they bind to the antigen antibody complex, and they brings about the killing. The last one is the lymphokine activated killer cells. They are activated by the chemicals released, which is known as the interleukins. And once they are activated, they kill the tumor cells, and they all do not display the activity against the normal cells. So that's it for the